purpose of this video is to give you a overview and reminder of things that you learned in general biology, maybe even back to high school biology. These won't be on the test for Unit 2. However, they are essential and critical for understanding many of the concepts that will be on the test. So hopefully this is review, just a reminder of things you've already know. If not, that's fine. You can review them and go over them here. So this is a diagram here of a protein, DNA polymerase in blue and purple, and DNA in the green and orange colors here. DNA is a double-stranded molecule. It's a very long molecule, and when you look at that long string, it seems very, very complex, but it's made up of fairly simple basic building blocks. So DNA is double-stranded. What that means is it has a strand on one side that is attached to a strand in the other side, and in a lot of ways you can think of it like a rope ladder with two sides and then the rungs going across. And if you took that rope ladder and then wound it up, it would make the helical structure, this so-called double helix that makes up DNA structure. DNA is directional, and what this means is the strand on one side is put together in a pattern that goes in a certain direction, and the matching strand on the other side goes the opposite direction. The orientation of those molecules is almost like a mirror image of one another. And the basic backbone of what we're looking at in the sides here, the sides of the so-called ladder of DNA, is very simple repeated molecular structure. So there's a phosphate group, there's a sugar group that forms the backbone for DNA, and from one element of this, these are called monomers, but from one element of DNA to the next one, to the next one, to the next one, they're all identical. And so we've got a strand and then an anti-parallel or an anti-sense strand on the other side. Now, the component of DNA that is not identical are what we're going to call the bases or the base pairs in between. And there are 4 different base pairs you need to know. Adenine, which is highlighted here in green, thymine, which is in this purplish pink color, guanine in blue, and cytosine in red. And you should just be familiar with those names. Also, you need to remember that those names are typically abbreviated by just the first letter of the name. So we have A for adenine, T for thymine, G for guanine, and C for cytosine. And that brings up another important point. You'll often see DNA and other biological molecules represented in different ways. You can see them in this stick model like this. You can see them in line drawings like the base pair here, or the kind of flattened out double-stranded piece of DNA up here in the upper right hand corner. We'll also be looking at space-filling molecules, which use balls to represent the individual molecules and put them together in ways that to some extent would replicate what they would look like if you could see them with your naked eye. Now, even though there are only 4 basic base pairs of DNA, this makes up for massive potential for diversity. So for each base pair, there are 4 different possibilities, and this can be represented mathematically by the equation 4 to the nth power, where n is the number of base pairs. So if you have one base pair, go back there. If you have one base pair, you have 4 possibilities, A, T, C, and G. If you had 2 base pairs strung together, you would have 4 to the second possibilities, or 16. So for instance, you could have AA, AT, AG, AC, and then so on through all the different combinations. And so what this means is by the time you get to 10 different base pairs in a row, there are over 1 million different unique possible strands of DNA that you could have. And by the time you get to 137 base pairs, which is a fairly short sequence of DNA, we have 3 times 10 to the 82nd possibilities. So this massive, massive number, more than or around the number of molecules, atoms, estimated to be in the entire visible universe. So it really is this huge possibility for diversity, and that's a key concept for understanding how DNA works as the genetic instruction manual for organisms. So again, review. These are space-filling molecules. Notice they look quite similar, although there are some differences. The primary difference is that The A and the G molecules are a little bit larger. They have an extra ring of molecules that the C and the T don't have. But notice they're quite similar but different enough that they are unique and can form the backbone for this genetic code. And again, sometimes you see these ribbon-like diagrams representing DNA. Other times you'll see the space-filling molecules, and you might even see stick molecules. But no matter how it's drawn, it's all supposed to represent the same thing. From here on out, mostly what you will be seeing are simply the letters. And just remember the letters represent those differing bases or base pairs, those molecules that provide the diversity for DNA. Now, when DNA is being copied, it's a very complicated process, and there are many, many enzymes involved. You learned about these enzymes in Bio 1. You may have even memorized some of the processes. For this class, we're not going to worry about the mechanisms. We're going to worry about the outcome and the results when DNA gets copied, and particularly when mistakes are made. So, although you might remember and know helicase, topoisomerase, primase, the different types of DNA polymerase, there are a variety of them and they vary slightly between organisms, ligase, gyrase, all of these molecules are critical for DNA replication. However, for this class, for these purposes, you don't need to remember any of them, any of that complicated stuff going on. It's really quite elegant and it's very dramatic and fast. This is a diagram showing how all these molecules fit together to copy DNA. And if you remember, you've got the leading strand, which can be copied directly, and then the lagging strand where the enzymes kind of have to jump ahead and then copy back so that they can fill in, and that's because of the directionality of the DNA. This is a beautiful animation that was put together to show this in process, all of these enzymes working together to copy DNA. Now, DNA replication like this occurs when cells are getting ready to divide. And this DNA replication occurs in mitosis, and it also occurs in meiosis. We'll look at the differences between mitosis and meiosis later on in the course. But for now, all of this complicated stuff we can simply represent as a series of letters, A's, T's, C's, and G's, that are being copied to make an exact version. But occasionally mistakes are made. Mutations occur. And so although DNA replication is very high fidelity, it's very rare to make mistakes. Some mistakes do slip through. And so mutation is typically thought of as a negative thing, right? So I think nowadays maybe there are two kind of immediate associations that people have when you say mutation. Number one is a mutant, an organism that's deformed or maybe not able to survive. Nowadays we kind of also have this popular idea of mutants as superheroes, right? The X-Men or Spider-Man or some sort of a mutation that gives somebody a superpower. So those are kind of two very extreme different views. Not all organisms have the same mutation rate. Most eukaryotic organisms have relatively low mutation rates with very, very rare mistakes. Other organisms, such as viruses, might have a much higher mutation rate. So these organisms have adapted to have mutation rates that allow them to do well long term in the niche that they are filling. But even if it's a much higher mutation rate, like in viruses, overall it's still a very rare event to have a mistake made and put a base pair in incorrectly when we are copying our DNA. So here is Dr. Terry's overview of mutations and the impact that they have on organisms. And I think this is a really good conceptual overview of the process. The numbers are my own. I don't have any data to back them up, and they might be much, much larger or a little bit smaller, but the idea is very solid. So don't quote me on the numbers, but the idea is very solid. So this is a pie chart representing all of the mutations, all of these mistakes that occur during the copying of DNA. And before we talk about the percentages and the numbers, it's also very good to remember that by and large, mutations are random mistakes. So imagine you're copying out a large manuscript, say the entire Oxford English Dictionary, and every once in a while, as you were transcribing it and copying it out, you put the wrong letter in, and it was just a random mistake. There's no pattern. You didn't do it more in one part of the dictionary or in the other. It was just random mistakes. And so it's good and a useful model to think about DNA mutation as a simple random mistake that occurs now and then, albeit somewhat rarely, as you are copying your DNA. So, of all those random mistakes that occur, a huge percentage of them, so here for illustration purposes I put 95%, but a vast majority of these random mistakes are effectively neutral. And what that means is the organism that carries that new mutation is not better off or worse off because it carries the mutation. It's essentially the same as it was before, although that mutation can be measured and found in their DNA. It does not impact the phenotype of the organism. Okay? We will talk about reasons for these percentages in 2.2. Of the remaining mutations, the vast majority of those remaining mutations are negative, or the technical term is deleterious, meaning that they have a negative impact on the fitness, the surviving, or the ability to reproduce of the organism that carries them. Now, those neutral mutations, the 95%, are not subject to natural selection, because natural selection works to either remove negative mutations or make beneficial mutations, positive mutations, become more and more common until they're spread throughout the population. So these 95% of neutral mutations have no force of natural selection working on them. But these 4.99% of deleterious mutations have strong natural selection working against them. And depending on the environment, the genetics, how many different alleles are there and the impact of those alleles, and perhaps even linked genes and other genetic factors, there's going to be various rates of removal of these negative mutations from a population. But they pop up by mutation, and the natural selection works to remove them from the population. So this white line here, this little tiny sliver on our pie chart, represents the remaining, maybe about 1 in a million or so, of random mutations that are beneficial and actually help the organism. And so a beneficial mutation, oops, I've changed this. Let me fix that real quickly. That's going to bother me. I made the mistake. That should be adaptive rather than. There we go. So that last 0.1% is adaptive. 0.0001% is adaptive. And again, maybe 1 out of a million. So most mutations are effectively neutral. The remainder of the mutations are, most of them are deleterious or negative, and it's only this little tiny fraction of mutations when they occur are actually beneficial. Okay? Now, after these mutations occur, genetic drift and natural selection work on them and change those overall percentages. So when we come around and look at organisms, we don't see this number quite like this. Still, a great majority would be effectively neutral of the variety that we see in organisms. We would not see nearly as many deleterious mutations because they've been removed and worked on by natural selection. And the small percentage of mutations that occur that are beneficial, we would see many, many more of these because natural selection works to push them to become much more common and pushes them to be dispersed throughout the entire population. And so remember, this pie chart just simply represents the mutations as they occur, not what is their final state and what occurs when we look at populations today, which is looking at mutation after they have been filtered through the forces of genetic drift and natural selection. So hopefully that's a little bit of a review for you, a little bit of a overview. And as we begin talking about mutations and genomes and how DNA works on organisms, this should provide a good foundation and background for many of the concepts that you will need to know for Unit 2.2.